Hello everybody, and welcome to PathoLogic. Today we're going to be talking about immunity and immunodeficiency. The immune system is key for preventing foreign pathogens from entering and proliferating within the body. There are two main systems involved: innate immunity and adaptive immunity. Failure of these systems results in immunodeficiency. We will start by briefly reviewing how the immune system works, beginning with innate immunity. Innate immunity provides an immediate defense against pathogens and helps eliminate damaged cells. This type of immunity does not remember previous pathogens, nor does it specifically target any given antigen. It simply reacts to anything unusual that it may find. Epithelial cells are an often forgotten component of innate immunity. These cells line the parts of the body that interact with the environment, such as the skin, gastrointestinal tract, and the lungs. They act as a mechanical barrier to prevent entry of pathogens, as well as produce antimicrobial proteins called defensins. Phagocytes detect pathogens and ingest them for destruction. The monocyte macrophage system is one of the primary phagocyte groups. In this system, monocytes migrate into the tissue and mature into macrophages. Neutrophils are also able to phagocytose pathogens. They are a bit more diverse in their capabilities, though, as they can also kill cells by releasing cytotoxic granules or by forming neutrophil extracellular traps that ensnare pathogens. At this point, you might be wondering how these phagocytes recognize pathogens in the first place. Cells of innate immunity primarily recognize components of microbes called pattern-associated molecular patterns, or PAMPs. These are typically components of the microbes that are essential for infectivity, so they are highly resistant to mutation. This provides a relatively consistent molecular pattern that phagocytes can recognize as foreign. PAMPs are recognized by pattern recognition receptors on the phagocyte. The two main categories of PRRs are toll-like receptors and nod-like receptors. These receptors are found on the plasma membrane, within the cytosol, or within endosomes, depending on their type. Plasma membrane receptors detect extracellular microbes within the interstitium. Cytosolic receptors detect any intracellular pathogens, and finally, the endosomal receptors detect ingested microbes after phagocytosis. Toll-like receptors are a major pattern recognition receptor for phagocytes. Mammals have 10 toll-like receptor types that each recognize different sets of microbial molecules. They are primarily found on the plasma membrane and in the endosomes. Activation of a TLR results in secretion of cytokines, production of interferons, and expression of adhesion molecules that help leukocytes migrate towards a site of infection. Nod-like receptors are found within the cytosol and recognize microbial products or products from necrotic or damaged cells. These receptors activate the inflammasome, resulting in activation of tissue-damaging caspases and production of interleukin-1, which is one of the major triggers of fever. Next up for the innate immunity participants are the dendritic cells. These cells are found in and around epithelia, lymphoid organs, and many other tissues. Their main role is to capture antigens and display them to T lymphocytes, resulting in activation of adaptive immunity. These cells also release cytokines upon activation. There are a few types of innate lymphoid cells, but we primarily think of NK or natural killer cells within this category. These cells reside in the tissues and they recognize stressed cells. These cells that are stressed could have viral infections or intracellular pathogens, or they could be cancerous cells that are trying to form a tumor. NK cells can also be activated by the cytokines that are released during tissue damage. The unique feature of innate lymphoid cells is that they do not require antigen presentation to become active, unlike most immune cells. They are also able to recognize and target cells that are missing MHC1, which we will discuss a little bit later. They are the only cells that are able to do this, which makes them useful for identifying damaged cells that would otherwise escape immune system recognition. Once activated, these cells produce similar cytokines to CD4 T cells. Innate lymphoid cells recognize damaged or stressed cells by identifying damage-associated molecular patterns, or DAMPs. When a cell is damaged, it increases expression of DAMPs and downregulates normal receptors, such as MHC1. On the NK cell, receptors that activate that cell bind to DAMPs and trigger a response. This allows them to target a damaged cell. However, NK cells also express inhibitory receptors. These receptors bind to things presented by MHC1 on healthy cells. This prevents activation of NK cells against normally functioning cells. Once NK cells are activated, they release cytokines like interferon gamma that help activate macrophages and other cells. NK cells can also be activated by interleukins that are produced during an immune response. For example, IL-2 and IL-15 can activate proliferation of NK cells, while IL-12 activates NK cells to begin killing target cells and secreting interferon gamma. Finally, in our innate immunity players, we have the complement proteins. The complement pathway is activated by microbes, and the end result of complement activation is formation of a membrane attack complex, or MAC. This complex basically forms a hole in the microbe membrane, disrupting its homeostasis. Complement is a very complex system that will likely be a topic for a future video, but for right now, just know that the alternative and lectin pathways are activated by innate immunity, while the classical pathway is used for adaptive immunity. So to summarize innate immunity, this system can target extracellular microbes through neutrophils, macrophages, and complement proteins, or it can target stressed cells through its NK cells. Activation of the innate immune system ultimately activates the adaptive immune system through release of cytokines, interleukins, etc. Antigen presentation by dendritic cells has a specific role in activating adaptive immunity by preventing the specific target antigen to adaptive immune cells. Now we will review how the adaptive immune system works. The adaptive immune system has two basic components: humoral immunity, which is governed by B cells and their antibodies, and cell-mediated immunity, which is governed by T cells. The key activators of adaptive immunity are antigens that are presented by another type of cell. Cells present these antigens on proteins called major histocompatibility complex, or MHC, which is on their cell surface. There are two main types of MHC, MHC1 and MHC2. MHC1 is expressed by all nucleated cells, as well as platelets. They display peptides derived from cytoplasmic proteins that the cell has produced. So basically, they're showing the immune system, hey, I just made this thing, and the immune system evaluates it to see if it's abnormal. MHC2 is expressed on macrophages, B lymphocytes, and dendritic cells. This MHC presents antigens that the pattern recognition receptors on the cell bind to. These PRRs can be on the plasma membrane, in the cytosol, or in the endosomes, as we discussed previously. This MHC allows these cells to show the immune system what they found during their travels. Unlike the innate immune system, the adaptive immune system is highly specific. It only activates against one particular antigen. This is possible because of high lymphocyte diversity. Within the body, there are millions of lymphocytes that are each looking for a different antigen. Estimates suggest that the average adult has lymphocytes that can recognize between 10 to the 7 and 10 to the 9 different types of antigen. If a lymphocyte finds its specific antigen, it undergoes a process called clonal selection. This is the process of proliferating numerous genetically identical daughter cells from that one cell. Because these cells are genetically identical, the new cells also target the specific antigen of its parent cell. This is the basic foundation of adaptive immunity, as basically once the adaptive immune system recognizes a specific antigen, it can produce millions of lymphocytes that are targeting that particular invader. So now we will discuss the major players in adaptive immunity. These are the T cells, B cells, dendritic cells, and macrophages. There are two major types of T cells. Helper T cells stimulate B lymphocytes and activate other leukocytes. Cytotoxic or killer T cells kill infected cells that the immune system identifies. These cells develop in the thymus before migrating into the blood and secondary lymphoid organs. T cells recognize specific antigens via their T cell receptor. The cell will also express CD4 or CD8 proteins depending on its job. CD4 positive cells are helper T cells, while CD8 positive cells are the cytotoxic killer cells. These proteins act as a co-receptor for T cell activation to help ensure that the immune system is only activated if necessary. CD4 binds to MHC2 on antigen-presenting cells, like dendritic cells, after the T cell receptor binds to its antigen. This allows for activation of that helper T cell. Once activated, these cells secrete IL-2, which helps stimulate T cell proliferation, and it also begins expressing CD40, which can bind to ligands on macrophages and B cells to help activate those cells. CD8 binds to MHC1 on cells that are presenting antigen to cytotoxic killer cells. The activated CD8 cell then releases granules that contain perforin and granzyme. These compounds trigger apoptosis in the affected cell, thus leading to cell death. Next, we have the B cells. These are the cells that produce antibodies. They develop in the bone marrow and then migrate into the blood, lymph nodes, spleen, etc. They are primarily activated by recognizing antigen bound to their B cell antigen receptor complex. However, they can also be activated by complement proteins or by helper T cells. When a B cell recognizes an antigen, it endocytoses it and displays it on MHC2. The presented antigen is recognized by a CD4 helper T cell, which becomes activated and expresses CD40. Expression of CD40 allows the helper T cell to activate more B cells. Once activated, B cells mature into a plasma cell and begin secreting antibodies against their target antigen. Secreted antibodies bind to microbes and prevent them from infecting the cells. Additionally, a specific type of antibody called IgG can opsonize the microbes by coating their surface. Opsonization activates phagocytes to consume the pathogen, so essentially IgG makes the pathogen more tasty to the phagocytes. IgG and IgM can also activate the complement system for pathogen killing, as well as for promoting phagocytosis. Some of the activated B cells are going to form memory cells. These cells do not produce antibodies during the initial infection, but instead proliferate within the germinal centers and enter the bloodstream. The purpose of these cells is to begin a rapid, antibody-driven response if that antigen reappears. These memory B cells can last for decades within the bloodstream and are the basis for vaccination. I mentioned a couple of antibody types already, so you may be wondering what antibody types do what. The type of antibody produced by a B cell largely depends on the type of antigen that it wants to target. For example, lipid and sugar targets bind best to IgM, while proteins bind best to IgG, IgA, and IgE. During the immune response, isotype switching allows the B cell to change what type of antibody it is producing. This process occurs in the germinal centers of lymph nodes, under the guidance of helper T cells. Now, we already talked about dendritic cells, but they also play an important role in adaptive immunity. This is because they are usually located under or around epithelia, which is a common site of microbial entry. They can also be found throughout many different tissues where antigens or infections may develop. Once these cells bind to an antigen, they migrate to the T cell zones of the lymphoid organs. This allows them to present the antigen to helper T cells, activating adaptive immunity. Macrophages also help play a role in adaptive immunity as an effector cell. These guys are going to phagocytose and destroy microbes and antigens after the target has been opsonized by IgG or by complement proteins. Once they phagocytose an antigen, they present it on MHC2 to the helper T lymphocytes. This further activates the adaptive immune response against that particular antigen. Now we will go through a basic immune response to help you guys put it all together. To start, a dendritic cell in the tissue identifies and phagocytoses a bacterium. The cell then carries the antigen to its local lymph node. It presents the antigen on MHC2 to a CD4 helper T cell. The helper T cell releases IL-2, which helps stimulate T cell proliferation. Activated helper and cytotoxic T cells then leave the lymph node and go to the site of injury. The CD4 helper T cells activate macrophages in the area through CD40. They also release cytokines that can activate unbound macrophages. The CD8 cytotoxic T cells can identify foreign antigen presented by stressed nucleated cells that are in the area and induce apoptosis in those cells. Activated CD4 helper T cells that are back in the lymph node are also going to express CD40, which can activate B cells. Those activated B cells release antibodies against the target antigen. Those antibodies enter the bloodstream and go to the site of injury, where they opsonize the target pathogen. The macrophages that were activated by the helper T cells then phagocytose that pathogen. Finally, back in the lymph node, there is also going to be production of memory B cells that are going to be released into the bloodstream. These cells are primed to target the same antigen if the body is re-exposed to that original pathogen. Now that we have a basic understanding of the immune system, we can discuss a few immunodeficiency disorders that occur in veterinary medicine. Immunodeficiency disorders occur due to a lack of function in some component of the immune system. This prevents an appropriate immune response. Typically, these immunodeficiency disorders manifest as an increased risk of infection or increased incidence of tumors. If you are wondering about the tumor risk, remember that NK cells and CD8 T cells can target stressed cells, which includes cancer cells. If these components are deficient, the body can no longer target and eliminate these early neoplastic cells. Finally, know that immunodeficiencies can either be primary or secondary. We're going to start by discussing some primary immunodeficiencies. These occur due to a congenital or genetic defect, and there are several of them described in veterinary medicine. These conditions can affect components of either the innate or adaptive immune system. We'll start by looking at a few conditions that affect the innate immune system. C3 deficiency is the most significant complement pathway deficiency in mammals, as it affects all three pathways of complement. This condition has been described in dogs. It predisposes the animal to bacterial infections, as complement primarily targets those types of pathogens. Shediac-Higashi syndrome is a disease that affects the release of granules from cells throughout the body. In the immune system, this prevents neutrophils, NK cells, and cytotoxic T cells from releasing their granules that they use for cell killing. It can also affect cytokine release from cells, preventing appropriate signaling between immune cells. The combined effect of these factors results in an increased infection risk for the animal. Next, we will look at some conditions affecting the adaptive immune system. Severe combined immunodeficiency is a condition that primarily results in non-functional T lymphocytes. However, because there are defective helper T cells, this can also prevent appropriate activation of B cells, potentially resulting in a limited B cell response. Severe combined immunodeficiency also affects B cell functionality, such as the release of antibodies. The combined effect of these two deficiencies increases the animal's risk of viral, fungal, and bacterial infections. Although this condition is best diagnosed using histology and genetic testing, there are a few gross lesions that may help you identify an affected animal. These animals typically have a small thymus, as there is minimal T cell production in this disease. The spleen may also have minimal white pulp, and the lymph nodes may also be smaller than expected, both due to low numbers of lymphocytes. Thymic hypoplasia is a similar condition where there is a non-functional thymus. This results in a T lymphocyte deficiency. Similar to severe combined immunodeficiency, T lymphocyte deficiency can affect B cell activity. Therefore, these animals are more susceptible to many different types of infections. Common variable deficiency is a defect in B lymphocytes, resulting in minimal B lymphocyte production. Consequently, there is also minimal production of antibodies because there isn't enough B lymphocytes to produce them. In humans, this condition can occur from many different mutations, hence the variable part of the name, but all of those mutations lead to the same result of a B lymphocyte deficit, which is referred to in the common part of the name. Agamma-globulinemia is a condition where the B cells are unable to produce antibodies, which are also known as immunoglobulins. This condition is typically caused by genetic mutations that stop lymphocyte development, preventing B cells from developing into plasma cells that are able to produce antibodies. Therefore, affected animals will have no plasma cells or circulating antibodies on blood work. Finally, we have immunoglobulin deficiencies. These are caused by genetic mutations that result in a deficiency of a specific type of immunoglobulin. However, the B lymphocyte levels in these disorders are normal. IgM deficiency has been described in horses and can be associated with recurring respiratory infections. IgA deficiency has been described in dogs, where it causes recurring infections of the mucosal surfaces, because IgA is primarily used for mucosal defense. Finally, we have secondary immunodeficiencies. These occur due to some insult to the immune system during the animal's life. Here are a few example causes of secondary immunodeficiency. Pathogens can cause immunodeficiency by depleting lymphocytes or by affecting their maturation. A few examples of this include feline leukemia virus and feline immunodeficiency virus. Radiation therapy or chemotherapy can also affect the immune system by decreasing bone marrow precursors that are available for leukocyte production. Bone marrow neoplasia can prevent leukocyte development by reducing the available area within the bone marrow for producing lymphocytes, as well as reducing precursors. Malnutrition can prevent formation of antibodies due to the lack of protein within the body, which is one of the main building blocks required to produce antibodies. Finally, failure of passive transfer results in no antibodies in the body due to a lack of colostrum ingestion. This is considered a secondary immunodeficiency because the young animal's immune system is intact without any genetic abnormalities. They just did not receive what they needed from their mother's colostrum. To summarize, immunodeficiency can result from defects in either the innate or adaptive immune system. Innate immunity includes cells like phagocytes, dendritic cells, NK cells, and complement proteins. Adaptive immunity is primarily regulated by B and T cells. Of the immunodeficiencies, secondary immunodeficiencies are much more common and include things like failure of passive transfer or virus-induced immunodeficiencies. Thank you for watching PathoLogic. If you enjoyed this video, remember to like, subscribe, and to share with your classmates. It really helps the channel grow and reach more vet students. Thanks again for watching, and we will see you in the next video.